that's published recently by Manil Estelar. So obviously epigenetics is very important, but what I will also argue is that not only epigenetics is now recognized as a hallmark by Hanahan, because without that you can't call it a hallmark. Epigenetics actually is involved in every hallmark, right? And I think 10, 15 years ago when I was mentioning, that was not very clear, not very accepted, but I think now we can easily accept metastasis, autophagy, immune evasion. In all processes, epigenetics is involved. So not only is a hallmark, it's also a phenomenon that drives, or at least contributes, in all the other hallmarks of cancer. Now, we have seen this slide before, or not this slide, but the fact that 90% of the cancer-related death happens due to metastasis. Sorry, the pointer. That works, right? Yep. Slide forward doesn't work, but pointing works. So 90% of the cancer-related death happens due to metastasis. And it still remains unknown, relatively unknown, why it is that primary cancer cells become metastatic. And Vogelstein says that another possible explanation is there is no gene that is responsible for metastasis. So the question you can ask that the primary tumor, which is already harboring all the driver mutation, BRAF, NRAS, P53, whatever, ABC, name it, they already have it. So surely they are not the one which is driving the metastasis process alone. It must be something else that is happening that is actually driving. What is it? So we are working on this hypothesis that epigenetic events play a driver role in metastasis of cancer. Now, it's very difficult, actually, it's not been established, how would you identify epigenetic drivers? The driver of genetic drivers are fairly well described, but how do you describe epigenetic driver? And the framework was not established. So we have provided some ideas, and actually it's been very well cited and accepted as epigenetic driver. So you have this normal cell, you have epigenetic changes or genetic changes, it accumulates. And then if you have a selective epigenetic changes, so DNA methylation in this case, that means you give a selective advantage, those go on to become metastatic clones. And epigenetic changes are dynamic, it is reversible, it allows cell to change the way they want to be, whatever they want to be. So it makes perfect sense that epigenetic changes provides that driving factor for a cancer cell from a primary organ to go to brain, go to liver, go to wherever and colonize and metastasize. So I'll show a few examples on that, on colorectal, and quickly move into quite a few other things. This is a work in colorectal cancer we have done where we have profiled normal colon, primary colon, and metastatic colon. Now, these are matched patient samples. So the metastatic liver met, they are derived from the same patient. So it's absolutely normal, primary, metastatic. So we actually have less heterogeneity, a direct comparison. And you can see that we have identified really stark methylation changes. So this is 0 to 100 scale methylation changes between these three groups, which is quite extraordinary and shows, I'm not showing you the details, but shows it's very liver metastatic specific. This is a really nice independent validation we saw here. This is from PCGA. Obviously, we have done sequencing based work, so when we compared the common probes, we saw excellent validation in primary tumors from other cohorts. Now, the stunning thing is that when you put into a PCA with the 244 sites in the genome, you can completely segregate LM, that is liver met, from the primary tumor, from the normal colon. So this gives a signature to segregate liver mets. Now, going one step further, we have now added aberrant crypt, so crypt cells in the colon, which actually often gets oncogenesis, primary tumor from a different cohort, and also crypt foci, so colonic crypt. And with all that, we still are able to segregate liver metastasis from anything else in colorectal cancer. And also, these patients has right and left mix. Half of them were treated with chemotherapy, half of them were treatment naive. So it doesn't seem like anything is affecting this particular 244 signature that can distinguish liver mets. When you did transcriptome on the same patients, we have done transcriptome, matching transcriptome. Transcriptome shows a large change but cannot differentiate the way DNA methylation did. It seems methylation is very programmed and not stochastic. Here are some new data This is on early changes, early drivers. So metastatic primary tumor, lymph node. So the first part where the tumor goes, early stage of metastasis, and these are normal colon. And again, you can see we've identified some really dramatic changes between this primary normal colon, primary metastatic. And if you see some of these changes, they are really dramatic, like 100% coming down. So it's progressive changes, some progressive hypermethylation, some stochastic changes as well, but very discriminatory changes, even at a very early stage of metastasis. We also have identified, done decades of work and identified driver, potential epigenetic driver. So here's an example of EBF3, which you can see in all the cells that we looked at. They are normal cells in green color. They are completely unmethylated. And any cancer we looked at, it's very, very highly methylated, that's 100%. So we showed that it's possible to actually identify pan-cancer methylation driver for metastasis, or a universal marker of metastasis. Now, just changing a tack a little bit, going to a little bit of basic biology, we looked at expression of this gene. Now, when you have a hypermethylation in the promoter, what do you expect? The gene will be silenced, right? But we saw opposite. We saw an increase in expression with the hypermethylation. So then we did an azacitidine experiment. We removed methylation globally, and the expectation was that it will actually reduce because it's opposite, right? And exactly that's what we saw. So you treat it with azacitidine, which is global method, and the expression goes down. So we know that high methylation shows high expression, but we still did not know how and whether it's really causal or whether it's a consequence effect of some other things. So we really need to develop a tool that is CRISPR editing or epigenomic editing. So basically what you do, you have a dCas9, so dead Cas9 that doesn't change the sequence. You have a target gene to take your guide RNA, and then if you can remove methylation basically, or you can add methylation, whatever you want. You need to develop two systems for adding DNMT3A, for removing TEP system, and then you can take on and off and then see what happens. So here's an example, another example, like a successful experiment. This is melanoma cell lines. We have added methylation. You can see they were really low. We were able to add quite a lot of methylation. These were demethylation, 40% here, 59%. We are completely able to wipe off methylation from that same locus. So different cell lines, we have done this. So it's beautifully worked. I'll show you another example. This is a heat map of high throughput sequencing. So this is the ABF3 target region. This was before editing, and this is after editing. It's completely gone. And it's not even patchy clonal, it's completely gone. So we're very pleased about that. The final test was that if you just do this, just change methylation and nothing else, no sequence is changing, nothing is changing, can it alter the EBF3 expression? That will be the ultimate test to say it's causal. And here it is, it does change. So, but remember, it's paradoxical, it's the other way down, okay? So when we removed methylation, then we saw, so this is unedited, and we saw increase. These are two different guide RNA. We saw a change in methylation, and then when you wiped off methylation from this line, which I showed before, you see decrease. So high methylation drives expression. We have clearly shown this. This is a qPCR experiment. We have recently done RNA-seq as well, and in a different line, and we confirmed the finding, qPCR finding, that high methylation drives high expression. With that, we were able to propose a framework, a new phenomenon, a new mechanism of paradoxical activation by DNA methylation, which now is quite accepted that it does happen. And we have proposed some mechanism how it happens. There are possible 3 mechanisms that it could happen via. So that was a sort of new phenomenon. So someone says methylation is silencing, you can challenge that it's always not the case. You have to taste it if that's the case. Okay, we're now developing screening, so CRISPR screening of methylation. So not one gene, we want to edit hundreds of them, because then we can actually alter phenotype, we can maybe reduce metastasis and so on. So that's something, that's the work is ongoing in the lab. I'll quickly jump into liquid biopsy now in the final, some sort of part of the talk. So we're now working on cell-free, also a liquid biopsy, so cell-free DNA. So it's a cell-free tumor DNA from the patient blood. It has tremendous potential of obviously transforming and translating. We have heard the talk from Prasant yesterday, and he gave some background to it, so I'll jump into this. Now, we've heard yesterday OMG, omics. I absolutely agree. It's very expensive, and when we think about translating things, then one thing is that you can do a lot of fancy stuff, and then if it costs $10,000 per patient, then let's be honest, it will never get into clinic. Particularly in New Zealand, it's a publicly funded health system, same in UK, it's NHS. So it's basically government, public funded. So if you develop a test, it might be amazing, cost $10,000, never ever it will get into clinic. So you really have to think about fast, cost effective, yet accurate, and better than the current treatment. Only then something will happen. So we are actually discovery phase, so we can't do like a $50 stuff, which we will be able to do. So we needed to choose a method. Now we are fortunate that we were able to work on this technique for decades. We were the pioneer of this technique, and we have heavily published. You can see, so it's a reduced representation. It's not whole genome. It enriches for functional regions in the genome, and analyze almost 4 million CPG, which are functionally important dynamic CPGs across the genome, across 20,000, 22,000 genes. So we have actually employed this technique, and this is the first multiplex example. A long time ago. This is our recent one on a clinical, how to adopt in a clinical biospecimen. So this is a technique we have now adopted for cell-free DNA, which is a next frontier of challenge. So really small amount of DNA, few nanograms, we could do an excellent correlation we obtain from those. And that's something we now are applying, we have applied, and I'll show you some results from this. Again, I'll quickly skip this because you can see very nice correlation between different platforms of bisulfite. So we are very pleased with that. This is a new technique we have tried, EMC, just putting it out there if you are interested. It's an enzyme-based new technique. In our experience, it doesn't work very well because the enzyme is variable. It's not as good as bisulfite sequencing. And here is the data to show that the technical correlation is not that great. So we are actually sticking with CFRDS, which is showing excellent correlation in the data. Because obviously, if it's going to make a clinical difference, it has to work every time, it has to produce same result every time. So we have embarked on a big project on colorectal cancer. The idea is to actually contribute in the screening space. So in colorectal cancer in New Zealand, people go through colonoscopy at the age of 60 to 74. And they get obviously an outcome. Now, a lot of people go through, and very few people get a cancer-related benefit outcome. Plus, it's obviously a painful process, and it costs a lot of money to the health system. And as a result, the screening process is not being extended to younger people because the health system won't be able to cope with it. So if you can have a blood-based test, it will be wonderful, and that's what we're trying to do. So the project is ongoing now. We're very excited. Just all the clinicians, gastro unit, everyone is working together on this. So screening population, FIT test is a fecal test. That's what they use in New Zealand at the moment, which the sensitivity specificity is quite poor, but it's easy. Anyway, so then the plasma sample, that's what we're taking here, and then obviously the patient going through colonoscopy. So we have collected 77 samples so far. We will be collecting hundreds, thousands of them in coming years, and the idea is to develop blood-based tests. So with the methylation-based test, we are hoping that this is what it could achieve. So currently, majority of the patients don't have any outcome, no cancer. Some has polyp, and only this percentage is cancer. So 78% in New Zealand. I don't know the Indian statistics, but I think it will be close for colonoscopy. Most people obviously don't have a benefit. And then the idea is with the triaging, that means a parallel adjunct test to sort of triage patients who doesn't need to go to colonoscopy. Hopefully it will become like this. So very less, no outcome, and then polyp, obviously that can be intervene, and cancer. So then people who are going through colonoscopy, if the test is accurate enough, then they will get the benefit, and that means the screening test could be expanded to a population that is currently not reached. That is 30-year-old, 40-year-old, and we also know that the cancer cases in young population, particularly colorectal cancer, is increasing, which has been mentioned. And here is a very, very preliminary data. This is non-malignant control. So non-malignant, I want to raise this point, this is not healthy control. And in a hospital setting, you'll never get a healthy person, like you'll have something, right? So non-malignant is diverticulitis, Crohn's disease, or IBD, or those kind of patients, but not cancer. And that's the one we need to really differentiate, not between healthy in the street versus cancer patient. Then in the real life setting, it's not going to work. So it's a non-malignant patient, early and late stage colorectal cancer. We're seeing some difference, but obviously we have to do a lot of patient samples. We also embarked on lung cancer, again a big project to identify early stage lung cancer. We're now embarking on immunotherapy related, also identify predictive marker and so on. And I'll show you a very quick data. It just came in last night. My PhD students sent it. 33 patients we have a profile so far, healthy, early stage lung cancer, late stage lung cancer. I'm showing you some top DMRs, and we're seeing some really interesting differences. And if you see the PCA plot, it's very discriminatory. So 33 patients, low number of samples we are carrying on. We have 200 patients so far collected. So we do all that and we get a really good picture. And as you know, in lung cancer, biopsy is very, very difficult. You cannot get lung biopsy. So it will be a game changer. Obviously, if we can have more and more blood-based tests, cheap, cost-effective, very highly sensitive and specific for lung cancer, for not only for diagnosis, but also for treatment monitoring, predicting, and so on. So that's the aim with the lung cancer. Prostate cancer, again, another big project. So all these are like co-developed with the clinicians and nurses, and this has been maybe my last 5 to 7 years of effort. It has taken a lot more effort to do this than the science, because to make a translational impact is really important. You have those teams so that you can get somewhere. And we did not think just identifying biomarker and publishing will actually get there. We really needed that link and network. So this is a prostate cancer project. We have obviously a lot of people, Maori investigator, clinicians, surgeons, oncologists, and name it. But I'll show you the data. Again, ongoing work, a lot more patients to be profiled in coming few years. So this is a healthy plasma, this is non-malignant. That means their PSA was raised, they came to the clinic, and then a biopsy was taken and they were non-malignant. Okay. So they actually went through with high PSA. And this is malignant PCA, so prostate cancer. They were malignant. So they got cancer detected. And you can see their Gleason score here, PSA and ISAP grade, which has been recently used quite a lot. So look at, I'll just give an example. This is actually obviously massive methylation difference, very impressive. But look at this last person. The PSA is 5, just made the threshold to be screened for biopsy. But look at the methylation profile, absolutely aggressive. And the Gleason is just 6, right? But it in the molecular profile is more like Gleason 8. So very, very aggressive. Now, in a normal screening setting, this person might be missed. Oh, yeah, 5 PSA, not too bad, and fine. But actually, molecular profiling will give a much better stratification, much more confidence in how to manage these patients. And here you can see 10.9. PSA raised, but they fall under non-malignant. So obviously, we know we've talked about this a lot. PSA is not a good marker, but here is example, and here is how DNA methylation can hopefully make a difference in future. We also have matched TRUST samples, a transrectal ultrasound tissue biopsy sample from the same patient. So we will also do those to see whether the TRUST biopsy is painful. They do 12 cores, and then they try to sort of slide it up and then actually try to see whether this cancer present or not. And it can be complemented as well with the blood test. Here is some new ways to looking into data. This is single patient 1, 2, 3, 4. If you look at one, we all average the sequence, right? We get a methylation value, and this is what I've shown you so far.